What a little girl. Kate, what a little girl's made of.
Get in here.
Sugar and spice.
Now we're going to put you in there.
That's nice.
We're going to blow you all to pieces.
Have you ever wondered really what it is that you are made
ready,
of?
Bill?
Yes, well, I'm going to ask for a
go.
volunteer for a very dangerous experiment. Someone who's going to be blown to pieces so we can find out what really is inside them. Do we have a volunteer anywhere? Volunteer? Volunteer. Oxygen. Here we've got one very brave volunteer. What's your name? Kate.
Potassium,
now will you get in
sodium,
here? Now we're going to put you in there. We're going to blow you all to pieces. Right? Okay. Okay, you ready, Bill?
Ready.
Yes, let's go.
The same elements that there are in the whole of the universe.
She's
what
all
else
blown to pieces.
could we be made of?
Let's see what was inside her, shall we?
Now then.
What was inside?
So
there are the elements
inside.
What they're
made of.
Hydrogen,
constituents,
oxygen,
and they
nitrogen, chlorine, some of the metals,
iron, potassium, sodium,
now,
magnesium, a bit of phosphorus.
Do you think she's going to be put back together again? The elements,
I hope
just
she will
the
be.
same elements that there
Because
are
of course living material
in the whole of the universe. What else
could we be made of?
I wonder what's happened to Kate.
Now then,
I hope we're going to get her back together again. You think we are? Do you think we are? Is she? Oh, is she coming? Where's Kate?
Here I am.
There you are, Kate. There you are. See, we've got it all back together again, haven't we? All together in one piece. Thank you very much indeed. Well, you see, the molecules that we've put her back together with, they don't exist as elements, as separate entities. They exist in combinations. And of course, one of the simplest of combinations is water. 2 hydrogen atoms and an oxygen atom. And I think we've got a little experiment here, Bill, just to show you how things happen. We've got some hydrogen and some oxygen in there, and we're going to make some water, right? We're going to make some water to show you how the simplest of those combinations works. Because molecules are combinations of atoms. Now let's see what's going to happen there. What's going to happen? We've made some water. Show them the water we've made. See, that's a combination of hydrogen and oxygen. Now, of course, there are all sorts of molecules, all sorts of combinations. Sugar. Sugar's a combination of molecules, isn't it? Sugar's a relatively simple molecule, but it's more complicated than water. It's got about 10 or 20 atoms for each of its molecules. And there are many different types of sugars that go up to make different constituents of living material. What's happening to that toast? I think it's burning a bit. Is it burning? Powdery stuff, that, isn't it? Just like coal dust. Well, it is really just like coal dust, isn't it? Because it's carbon, one of the other major constituents of living material. And sugars are made up of carbon, combinations of carbon, hydrogen, and oxygen. Now, I wonder whether we've got someone who'd like to do a little cooking. Have we got someone who'd like to do a little cooking? Where's a cook? Let's have a cook there. Yes, come on. Let's come down and what's your name?
Owen.
Owen. There we are. Well, put on your hat there. Put on your hat and let's do a little cooking. Right? Come over here. We're going to cook some bacon. Right? Put the bacon on there. Put it on the Bunsen burner. Put the pan on and let's put some bacon in there, shall we? Yes, good old bit of bacon. Bit late for breakfast. Never mind. Anyway, so we put the bacon in there, and what happens? The fat in the bacon melts out, doesn't it? Do you want to show them how the fat's melting out of the bacon? You're a pretty quick cook. Let's show them. You see? Just hold the pan up. You see the fat in there has melted away from the bacon. Because the fat is another molecule, another combination, this time mainly of carbon and hydrogen. That's what we call carbohydrates. And actually, then, when you've Now, we can't just live, though, on fats and sugars and salts. There's a bit of rock salt. Here's a bit of fat. Ooh, it's not very nice, is it? We can't just live on water, sugar, salt and fat, can we? We've got to have protein in the diet. Protein's an essential constituent of our diet. So let's see what's happened to that bacon now, shall we? Look at that. You see, when you've really cooked the bacon, what have you got left? You've got the meaty bits and the rind. That's the protein. So those are the proteins, the complex molecules. Now, I think we'd better have a little egg with our bacon, hadn't we? Do you want to just break an egg into there and let's see what happens. Show everybody how we're going to have some nice bacon and eggs. If you haven't had lunch, here's your lunch coming up now. Bacon and eggs. Just show them that. Show them how the egg white. There we are. You see, the white of the egg is another protein, albumin. And of course, the yellow part of the egg, that's full of proteins too. Well, that's excellent cooking. I hope you do as well at home. Thank you very much indeed for all your cooking. Let's put the pan back down there, shall we? Thank you very much, Owen. What a marvelous cook. Well, proteins are very big, complicated molecules. They don't just have 10 or 20 atoms like the sugars. They've got thousands of atoms. And the chemistry of living material is the chemistry of these very complicated molecules. It's those molecules that really are the spice of life. And it's the approach to studying living material, trying to break it down into its constituents, understanding how they're put together, that leads to our present remarkable understanding of living material and our ability to manipulate the genes, some of the things I'm talking about in these lectures. Now, there are many, many different sorts of proteins. We've already talked about some of them. And those proteins make up the structure of the body, and they carry out the functions by which the body works. Skin is a mixture of two main sorts of proteins, of collagen and keratin. The collagens are found in all sorts of different parts of the body, in the tendons, in cartilage, they're found in bone, they're found in teeth. Even an elephant's tusk has a lot of collagen in it. And of course you find the keratins, they're the other component of the skin, in your hair, and in nails, and in all sorts of in hooves of animals. Lots of different sorts of proteins. Meat. What is meat? Meat is muscle. And muscle has proteins in it, several types of proteins, which have to work together so that you can contract and relax your muscles. Milk, of course, that's protein, isn't it? A major component of milk is the protein casein. So we've got all sorts of proteins in the eye, in all the different parts of the body, many of them that carry out an important structural function. But there's another protein that's very important in the blood, and that's called haemoglobin. Haemoglobin is the substance that makes red cells look red. It makes the blood look red. And it carries out a most important function, that of carrying oxygen around the blood to the different tissues. Now, we've got a little experiment to show you how we can get out the haemoglobin from the blood. So we've got here a sample of blood. Here's some blood, real human blood. Now, how are we going to get the cells separated from the liquid part, the serum of the blood? We use an instrument called a centrifuge. Centrifuge whirls around like that. This is an old one. Usually you can't make them go nowadays when the lid's open. If we turn it around fast like that, then what happens when the centrifuge stops? The centrifugal force will have brought the red cells down to the bottom, will have separated them from the serum, from the liquid part, and we'd have a picture like this. You see the red cells at the bottom and the liquid part of the blood above them. So now we want to get the haemoglobin out of those red cells. So we take the liquid part away from the red cells, and what we're left then with is a sediment of red cells, a small number of red cells at the bottom. Now what we do in order to get the haemoglobin out of them, we add some water. Now water bursts open the red cells because they usually are not in water, they're in serum, which has the right salt concentration. Now what you'll see is, you see how that's going clear? Compare it with that, totally different. You see, the water has burst the cells open, and has released the hemoglobin from them. So now how are we going to separate the hemoglobin from the red cells that's come out from the red cells? We're going to add a little acid to that, and the acid will precipitate the protein just like you can curdle things with lemon. And you see the protein, the hemoglobin, precipitating out. Now we can separate that protein from the liquid part of the blood by spinning it down again in our centrifuge. And then what do we find? We find that. We find the protein, the hemoglobin, released from the red cells at the bottom, separated from the liquid part. So we can isolate that hemoglobin and study it. Now, hemoglobin has this all-important function of carrying oxygen around the blood. And we've got a little experiment to show you how that works. Thank you, Cynthia. Here we are. We've got some blood. And you'll notice it's a dark color. It's not really a bright red at all, is it? Now, what we're going to do is we're going to bubble some oxygen through this to show you what really happens to blood when it goes through the lungs. When the blood goes through the lungs, it picks up its oxygen there, and it turns bright red, and when it goes to the tissues, it releases its oxygen, and it goes dark again. It goes through a cycle of picking up oxygen in the lungs and releasing it again when it goes to the tissue. Can you see how that's turning bright red? You see that? As we bubble the oxygen through, the hemoglobin in the red cells is picking up its oxygen again, just as it would do if it was going through the lungs. Thank you very much, Cynthia. So hemoglobin has that all-important function of carrying oxygen to the different tissues in the body, the oxygen that they need to survive. But there are many other functional molecules, and a major class of the functional molecules are the enzymes. They're the workhorses. They're the things that do functions in the cell. They break down the food. They build it back up again into the constituents of the body from which we're all made. They can work chemical reactions with enormous efficiency, acting as catalysts. Now, we're going to do a little experiment here to show you just how an enzyme works. Can we have a volunteer here to do a little? Let's have you. What's?
I'm Madeline.
Madeline. Okay, Madeline. Now, we've got some liver here. You want to cut the liver up there. We're going to cut yes, you better use that nice sharp knife. Use the sharp knife. Cut the liver up and put it into that cylinder there. So what we're going to show you is how the liver has an enzyme activity that can break down things. Let's see if we can cut it up. Okay. Yes, maybe another piece of liver in there. We'd better. Okay. Now, while you're doing that, I'm going to pour in here some hydrogen peroxide. Now, hydrogen peroxide is a dangerous substance. And if we put that in there, why don't you go on cutting the liver up a bit, Madeline? Because I think it's not nice to have a lot of raw liver. See what's happening there? See those bubbles coming out there? See that? The liver is breaking down the hydrogen peroxide into water and oxygen. And what's bubbling off there is the oxygen, because hydrogen peroxide is a dangerous substance, and the liver is the organ that breaks it down. So there's an enzyme in the liver that does that most efficiently. I think that's enough. Now, why don't we take a little bit of that and let's cook it there, shall we? Because it's not very nice. You don't eat raw liver. Any of you eat raw liver? I don't imagine you do. So we're going to cook it there, and then maybe we can do something with it later. Just put some of the liver in the boiling water there, and then we can have some boiled liver later. Okay, there we are. Thank you very much indeed, Madeline. Thank you for that. Well, what really are the proteins? How can there be so many different types of proteins? The way you study them is to break them down into their constituents. If you take a protein and boil it in a strong acid, you break it down into its basic constituents. And when you do that, you find that there's a set of chemical substances called the amino acids, a well-defined set that is in all proteins. Here's an example of a model chemical structure of an amino acid. It's called amino because it has an amino group, just like in ammonia. The blue is for nitrogen. And it's an acid because it has a weak acidic component, a carboxyl group. So here you've got a model of an amino acid. And when you break down the proteins, you find that there are just 20 different sorts of amino acids. And we've got models of some of them laid out along the board here. Just 20 different basic building blocks in the proteins, all with the amino group and all with this acidic group and with different other structures. And a key feature of the structure is, of course, that they do contain nitrogen. So nitrogen is another of the major elements that you find in living material. Now, what really are the proteins? How then do these amino acids come together in a protein? Well, the answer is that the amino acids occur in sequences. So you can have a sequence of amino acids. Let's put this sequence up here. A sequence of different letters of the amino acids, one after the other. I wonder whether we've got some more amino acid letters here that we could use. Now, because what we can show you is that there are 20 different sorts of amino acids. So 20 different sorts of a single amino acid. If we have a pair of amino acids, then there'd be 20 times 20, or 400 different pairs of combinations. And if we had 3 amino acids, 1, 2, and a third one there, then we'd have 8,000 different combinations of 3 amino acids. And so you can see that with different combinations of amino acid sequences, we can have an enormous variety of different sorts of proteins. So that's how we can have so many different sorts of proteins. But how does that sequence of letters in a protein come together to make it work? It doesn't just exist as a long string. What it exists as is a complicated folded structure. Now, we've got a little model, I think, to show you how that comes together. Now, the first thing that we should talk about is to say, how can we establish the sequence of a protein? Because it's the sequence that gives it its unique structure. And the famous biochemist Fred Sanger was the first to establish the sequence of a protein. He worked out techniques for doing it, and he did it for the molecule insulin, the molecule that's essential for maintaining the level of sugar in the blood. And he found a unique sequence of the 51 amino acids that makes up that protein, and this is 9 of those, the first 9 of those amino acids. So in that way, he could show that there really was a unique structure to each protein made up by its particular sequence. And so different proteins have different sequences. And those sequences come together in complicated shapes, and that's what I'm going to show you now. So here we've got, just think of them as a string of amino acids. They're actually jelly bottles, a string of amino acids. But they don't exist as a long string. They exist all crunched up together to form a structure, a three-dimensional structure that is made by the particular way that that sequence of amino acid folds. Not a very nice structure that we've got there. So it's the structure of a protein that gives it its function. And we've got an example of some very nice computer graphics that we've got from the Open University Computer Department of a molecule, lysozyme, which bursts open bacteria, a molecule, an enzyme that was discovered by Fleming, who discovered penicillin, and whose structure was worked out by Sir David Phillips here at the Royal Institution. Here you see another version of that model. And notice this cleft here, that's where the molecule carries out its function, where it breaks down the product that it breaks down in the surface of the bacteria. So you see an enzyme molecule has a very specific shape. And that shape is what gives it its function. And here we've got a little model to show you how that works. Here's our enzyme. And here, when I put it together again, is the molecule that's going to be broken apart by the enzyme. Now, there's a specific shape to the enzyme here. Here's the molecule with its two parts to be broken together, and here's the enzyme. When I put that shape on there, what happens? The shape just fits the molecule it's working on to bring it apart, and you can put it together again too. So it's the shape of an enzyme that makes it work properly and gives it its function. Now, what happens if we destroy that shape? Let's have another volunteer, someone. Yes, why don't you come down? Yes, you. You're at the end of the row. Anyone? Yeah, come down. And let's see here. We've got our jelly babies, right? What's your name?
David.
David. Now, David's got those jelly bottles, right? Just put them into there, and let's see what happens. Let's see what happens. Let's just turn it. You see them separating apart? Why don't you just stir them around a bit with that? Let's see what happens. Stir them around a bit with that. You see they're all going into a goo. That structure of that linear molecule that was made into a three-dimensional structure has just disappeared. Now we've got a little, let's do another little experiment here. We've been cooking this, come down to this side of the table, David. We've been cooking this, let's take this away, shall we? Now we've been cooking that liver. Why don't you take a bit of liver out, take some of the liver out from there. Can you get it out there? Can you get it? You got, let's see, wait a minute. Have you got it? Get the liver. Have you got it? Piece there? Yeah, let's have a go. Let me have a go, and then we'll give it back to you. It's difficult to see in there, isn't it? Okay. Now, come along over here. Come along over here. Oh, let's put it on a plate there, shall we? Okay, let's get another bit out for you. No, stay here. Stay here. What I want you to do is now see if you. Maybe it's easier to use the fork. Let's put that liver. Put it in here. Bring it over here, David, and put it in here. Just put the liver in there. All right? Now, what we've got in there is some hydrogen. Put the other piece in too. Some hydrogen peroxide. It's not bubbling, is it? Not bubbling like this one is. Because we boiled the liver, we've destroyed the enzyme, it's not working anymore. Thank you very much, David. Thank you very much indeed. So, if we boil the liver, we destroy its structure, it loses its function. The enzymes lose their function. So the protein molecules are very complicated molecules. They can do all sorts of things, And at one time it was thought that one of the things they could do, let's have a look at our cell model again, is determine what the genes were. So in a cell it's doing all sorts of things, energy, structure, that's all the proteins. But remember, in the middle of the cell is the nucleus. And in the nucleus are the chromosomes. So to look for the chemical of which genes are made of, we should be looking at what's in the chromosomes. Now, there is protein in the chromosomes, but there's also another substance, another large complicated molecule in the chromosomes, and it's that substance which is the chemical that the genes are made of. Now, it was the Swiss chemist, Miescher, who decided that he wanted to find out what was in the nucleus of cells and what the chemical substances there might be. So he took pus from bandages that he got from a local hospital, separated the nuclei, extracted them, and looked at the chemical substances that he got. And he found one very important chemical substance that he called nuclein and that we now call nucleic acid. And we know that it's that chemical substance which is really the substance of which genes are made. And when you look at the chemistry of that substance in the same way that we looked at the chemistry of the proteins, break it down into its constituents, we then find that there are just 4 basic constituents of that substance, that nucleic acid. C for cytosine, G for guanine, T for thymine, and A for adenine. 4 different basic units. Not 20 like in the proteins, just 4. So that somehow the nucleic acids are made up of these 4 basic major constituents. Now, let's have a look, before we look at what the structure of the DNA is, let's have a look at how we can get DNA ourselves out of cells. So we've got a little experiment here to show you. In this flask, in this beaker here, we've got some human cells grown in the laboratory. And we're going to burst those open and extract the DNA from them. Now, the way that we burst the cells open, in this case, is we add a little washing up detergent. Cells don't like detergent, so just look at what happens when I put some of that washing up detergent in there. See how it goes clear and gooey? That's the DNA being released from the cells. Now, how can we separate out that DNA? It's very simple. We add a little alcohol, right? Time for a bit of Christmas spirit. There we are. We add a little alcohol. Now, what we can do there is we can fish the DNA that's been precipitated by the alcohol out of here. And we're going to show you just how pretty it looks. Now, Nigel's going to come up here, and we're going to switch on. That's a UV lamp, the sort of long-wave UV that sometimes makes your shirts look blue. And when we shine that on the DNA will fluoresce. It'll show up nicely. Now, let's have the lights down, can we? As we pick out the DNA. Let's have the lights down. We have the lights all the way down, and then we'll see the DNA shining there. That's the DNA that we've isolated from the center of the cell in just the same way that we would doing any experiments that we do in the laboratory nowadays. So there you see DNA extracted from a cell. Now, how do we know that DNA is really the material of which genes are made of, the inherited material? To go to that, we've got to go to the bacterium, the lowly bacteria which can be studied. And in the 1920s, pneumococcus was an important organism, a cause of pneumonia. And here we've got some pneumococcal bacteria growing on a plate there. And before antibiotics could deal with the disease, what we used to do is try and find weakened strains of pneumococcus that you could inoculate into people so that they would then be protected in future from further infections, something I'll be talking about in a later lecture. And Griffiths, a microbiologist, was working with these weakened strains and doing experiments inoculating them into mice, as one had to. So here's the original strain, the strain that's lethal, that gives you pneumonia. And here's a weakened strain. Now, when you put the living, virulent strain that causes disease into a mouse, what happens? It's a disease-causing organism. The mouse dies of pneumonia. But when you put the attenuated, the weakened strain in, the mouse survives. Now, if you take the virulent, the bad strain, and heat it, then, as you've seen, when you heat things, you destroy them. You put that heated strain in the mouse, it'll still survive. Now, what do you think happens if you put a mixture of the weakened strain that doesn't kill the mouse and the heat-killed strain that doesn't kill the mouse? Just mix those and put them into a mouse. Will the mouse survive? No answer? Will it? You sure? No, it dies. Because what's happened is that this killed strain has conferred on the weakened strain its property of the ability to kill. The dead strain has released something which has got into the living one and turned it from a benign into a dangerous organism. And Oswald Avery, famous chemist at the Rockefeller Institution, he repeated these experiments in the test tube. And he showed that the extract that one could get from the killed bacteria that conferred their properties on the living bacteria was DNA, was that substance, the nucleic acid substance. Now, it's interesting to reflect that that momentous discovery, a truly momentous discovery to show what the material is that genes are made of, had to be carried out using animals. Animals which died when you gave them the virulent bacteria. Later, it was possible to do these experiments in the test tube. But without those first experiments, we wouldn't now know about the chemistry of genes we wouldn't have the incredible advances that we now have in understanding of human living material. Now, the challenge is to find out what is the true nature of DNA. And what you can say is that DNA is some sort of a sequence of these different letters. But before we go on to discuss that, 4-letter sequence, rather like you've got 20 letters in the protein. But before we go on to discuss that, let's talk a little bit about the way that the structure of DNA, the 3-dimensional structure, was worked out. Watson and Crick were the ones who did it. Jim Watson, a young biologist and geneticist from the United States coming in the early 1950s to Cambridge to work with Francis Crick, an extrovert physicist, on the problem of the structure of DNA. And they solved it, and the story is told in a delightful way by Jim Watson in his book, The Double Helix, soon no doubt to be made into a film. And they told that story in a paper in the journal Nature, a short paper, you'll see an extract of it, a short paper of less than 1,000 words. A paper of absolutely fundamental importance that's changed our view of living material, that's more important even than Darwin's description of the theory of evolution by natural selection. Now, how did they achieve that? They had to work out the structure of DNA, and they did it using crystals. Crystals are also used to work out the structure of proteins. And you shine X-rays on the crystals, and a technique worked out by the Braggs, William and Lawrence, who are directors of the Royal Institution here, for which they got the Nobel Prize, allows you to take a blotchy picture like that, which you get from shining X-rays onto your crystals of DNA, and from that, using some complicated mathematics and computing, to really work out the actual structure in 3 dimensions of a molecule. And we've got a sample of the molecule here. Let's have a couple of volunteers. Why don't we have a couple of people on the front row there? Yes, do come down. Yes. What's your name?
Richard.
And another girl sitting next to you?
Susan.
Susan. Okay, now I want you to get hold of one side of the table and just do a little dance around. Get hold of the table and just dance around, turning it round. Now, what do you see? You see that that is a double helix. It's a right-handed twist. And the 2 helices are like the edges of a spiral staircase. And along the level of each step, you find the bases that we've been talking about. Thank you very much indeed. Thank you. Now, here you see a computer animation of the same thing, the helix turning round the 2 strands of the helix. So let's think a little bit more about the structure of DNA that comes there. Remember, each step has the bases in it. 2 helices, and those 2 helices are joined by 2 bases on their steps. And there's a very simple rule. Remember, A, T, G, and C are the bases. Opposite a T is an A on the same step. Opposite an A is a T. Opposite a G is a C. And opposite a C is a G. It's a very simple rule that gives each step the same size, and it means that when you've got the sequence along one side of the steps, the sequence along the other sides is very clearly determined. So there you've got the structure, the basic structure of DNA through those bases. Now, that structure also gives you a mechanism by which DNA can be replicated. An essential feature of the genetic material is that it must be able to duplicate itself. Let's see how that can happen. Supposing we have a string here on one of the 2 helices, a series of these letters. Then the other string must be a matching sequence, right? Opposite a T and A G a C, C a G, and so on. So that's our double helix, the basic constituents of it. Now, how can that duplicate itself? It's very simple. If it separates apart, its 2 parts separate, then along one strand, you can build the complementary strand. You can put in opposite a G a C, an A a T, opposite a C a G, and opposite a T and A. So you can build up new strands along the old to reform two daughter helices from the original one. And we've got a computer animation to show that here. See the helix turning. Here is its edge. Here are the steps of the ladders. You see there are always in two components of the steps, the bases that pair, separating out, and then the new bases being added. You see them being added one by one to form two new helices from the one old one. So that the structure of DNA really provided the answer to living material, to how a molecule can exist that copies itself. Watson and Crick, in their paper, said rather coyly, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." That's a good bit of scientese, isn't it? And there you see them standing beside their model. Francis Crick put it rather more succinctly in a letter to his son Michael. He said, "We think we have found the basic mechanism by which life comes from life." And so indeed they had. Now, how does that process of duplicating the DNA work? It needs enzymes. And Arthur Kornberg was the person who found that if you took an extract of bacteria, you could find enzymes in that which would carry out this duplication process. And when I went as a young scientist to Stanford University, I was working with his group to try and create life from life, to try and create the transforming DNA that one could use with bacteria. Well, we've got a little experiment here to show you how that duplication of DNA can be done in the laboratory. Nigel, do you want to come and give us a hand here? So what we have here are two tubes, each of which contain a little bit of DNA. And what Nigel's going to do, he's going to take out of that lead pot there some radioactive substance. They're radioactive bases, the constituents of the DNA. We use radioactivity as a tracer a great deal in our biochemical experiments. And he's going to add some of that carefully to each sample of DNA. And then the next thing we shall do is Nigel will add to one of the samples of DNA and not the other, the enzyme that Arthur Kornberg found could duplicate the DNA. Now what happens? You incubate the two tubes, one with the enzyme and the radioactive bases, the other without the enzyme and the radioactive bases. And we'd incubate those for a while. And then after the incubation, what's going to have happened is that the one with the enzyme in it should have copied new DNA from the old using the radioactive substrates. Now you see, when we do experiments like this with radioactivity, although it's a very low amount that's quite safe, we use gloves to protect ourselves and to stop contamination. So after the incubation has happened, what do? We add a little alcohol to one of the tubes, and then, just as we did in getting the DNA out from the cells, should we add a little more there? We isolate the DNA because it precipitates out. Can you see the strand of DNA there that we can precipitate out? Do you see it there? So we can do that from each sample, wash them to remove any of the radioactive tracer that hasn't been incorporated, and then find out which of the two samples, the one with the enzyme or the one without, has built those radioactive substrates into it. Just to show you, we've got radioactivity there in both of those tubes. But of course, we've carried out the experiment before, as we had to, and we've got a sample here. So Nigel's going to lift that up. You see, there's not much there. Let's have a look at the other sample. You see that? That's the sample that's got the newly duplicated DNA in it, that's made from the radioactive bases that we've added. Here in the laboratory, you see, here in the lecture theatre, as in the laboratory, we've made life from life. We've made new DNA from old, incorporated it to make new copies of DNA from old. It's a fundamental process that we can now do in the laboratory. Now let's think about the DNA and its structure and what it can do. The DNA has no obvious structure that carries out a function in the way that the proteins do. It just has a sequence of letters. And let's think about that sequence of letters. If there are single letters in the DNA sequence, there are 4 different types of single letters. Now, the sequence of the DNA, if it's going to be related to the sequence of the proteins, has to encode that language with 4 letters in it, has to encode a language with 20 letters into it to go from the DNA sequence to the protein sequence. 1 letter at a time isn't enough, only 4 combinations. 2 letters at a time would give you 4 times 4, 16 possible combinations. That's still not enough to code from the language with 4 letters to the 20-letter language of the proteins. But 3 letters, if we had 3-letter combinations, then that would be enough. 3-letter combinations would be 4 times 4 times 4, 64. Plenty to cope with the 20 different amino acids. So the code of life that takes us from DNA to proteins is a 3-letter code. There are 20 amino acids, so some of them have more than 3 letters to them. And there have to be stops as well. Now, we've got a rather difficult demonstration here, because it's a complicated process, to show you how that whole business of going from DNA to a protein works. And we've had to train up a couple of willing volunteers, so I wonder whether Simon and Madeline, you could come down here and join us. And let's go to the front here, where we've got laid out a bit of DNA sequence. So here's the DNA sequence from which we're going to make a protein. Now, actually, you can't just make it from the DNA. What turns out, the DNA is too precious. You copy it into another molecule, a very similar sort of molecule to DNA, another nucleic acid called RNA, which differs mainly in that instead of the yellow T's, we've got orange U's. But otherwise, the rules for making RNA are just the same as DNA. Copy it, opposite a C, a G, and opposite an A, a U, and so on. Now, Simon and Madeline are going to do this copying process now, aren't you? So let's put it on the floor first. Then we can all see what's going on. Take your sample of bases and start the copying from this end. So what they're going to do then is they're going to copy that DNA sequence using the pairing rules. Opposite a C, a G, opposite an A, a U instead of T because it's RNA, opposite a G, a C, and so on. A very simple pairing rule for going along that sequence. They're a double-headed enzyme copying DNA into RNA. And now it's that RNA which will be used to read off the code to make protein. And in order to do that, we've got to have some way of getting from a sequence of those letters, those 4 different sorts of letters, to the protein sequence of 20 different sorts. We've got to have a molecule that tells us which of the triplets corresponds to which amino acid. So at one end it's got the triplet, and at the other end it's got the amino acid that matches it. How are we doing making that sequence? Not doing too badly. So there's an adapter molecule that carries out that process of copying, of reading off the RNA 1 triplet at a time. And in a moment we're going to do that. Let's, just as Simon and Madeline are finishing off this sequence. So we're going to go all the way from the DNA through the RNA to making protein. How are we doing there? Nearly at the end? Nearly at the end? They're doing pretty well, don't you think? It's a complicated business, actually, being an RNA polymerase, being an enzyme that does that. Okay, isn't that splendid? Now, let's have this board, our protein synthesizing board. Let's lift it on here, and then Simon and Madeline, let's put your sequence, your RNA sequence, put it onto the board here. There we are. So we've made an RNA copy from the DNA, the first step in that process. Thank you very much indeed, Simon and Madeline. Now we come to the next stage of the process, and that's another very complicated stage. So we've had a couple of volunteers here, and Kate and Owen, I think. Kate and Owen are going to come down and help us do this. So let's stand here for a moment. Now, what we've got to do is we've got to interpret that using the code. Now let's hold up our code there. Now you see there Each triplet, green, green, orange, that's for glycine. Red, orange, orange, that's for isoleucine. Green, orange, orange, that's for valine. So that's our code. That tells us which triplet corresponds to which amino acid. Now let's have our tuba player, Mr. Tuba. Let's come in, because we've got to have a little music to do this complicated process. Well, why don't we all welcome Mr. Tuba here? So, we're going to go through this process of copying. So what you're going to do, we're going to use the ribosome, the machinery by which the copying takes place that moves along, and Kate is going to read off 3 letters at a time, and Owen is going to be the transfer molecule that transfers the amino acid onto the growing protein. So let's have a go.
Green, green, orange.
Okay, you see?
Red, orange, orange.
Each triplet corresponds to its amino acid, added one at a time.
Green, orange, orange.
We're not doing too badly, are we there? Moving along.
Green, red, red.
Okay. You see, each time, just 3 letters.
Blue, red, red.
And
Orange, green, orange.
see, 3 letters at a time, each time adding one of the amino acids.
Orange, green, orange.
Just 2 more to go.
Green, blue, orange.
Now we're nearly at the end of our little sequence. Just 1 more to go.
Orange, blue, orange.
There we are. Now we've made that sequence. So we've made the sequence reading off the code. Now let's bring the sequence up to the board here so that everybody can see it. Come round here and put the sequence on the board here, will you? Put the sequence up there. Do you notice anything about it? Looks familiar, doesn't it? It's the sequence of the beginning of the insulin molecule. Now, let's have another version of that whole tune again, Mr. Tuba player. Let's play the whole DNA sequence along. Isn't that a lovely tune? That's the music of the DNA. Thank you very much. Thank you, Kate and Owen. Thank you very much, Mr. Tuba player. So now you see how we've gone from the DNA through the RNA to the protein. Fundamental process of life, making the working molecules of the cell. If there's a mistake in the protein, then obviously something may go wrong. A disease, an inherited disease perhaps. Well, the first direct evidence for an inherited disease due to a difference in a protein came from looking at the protein haemoglobin. Herrick was a physician in Chicago, and he described a disease in the blood called anemia. There are many different sorts of anemia when there's a deficiency in the red cells that stops them from dealing with oxygen properly. And in this case, it was a very special sort of disease. It occurred particularly in blacks, American blacks. And he also found that when he looked at the blood of people with that disease, there was something very funny about it. And we've got that here on a microscope slide. And can you see the sickle cell, the sickle-shaped cell there? You see that sickle-shaped cell there? And you see others. And another one there. Those funny-shaped red cells. They were characteristic of that disease, and that's what gave the disease its name, sickle cell anemia. It's a disease that's a very troublesome disease. And let's see why. We've got some animation that shows what happens when red cells in such a person flow down the capillaries. Gradually, as the oxygen runs out a bit, they become sickled, those red cells, and sooner or later, one of them gets trapped, and then you'll see in a moment how it stops. See that? And you get a terrible problem. So sickle cell anemia is a very distressing disease indeed. Now, what is it due to? Linus Pauling, the great chemist, he argued that hemoglobin was a major component of the blood, carried the oxygen around. Perhaps the sickle cell disease was due to an abnormality in the hemoglobin. And indeed, he looked and found with his colleagues that people with the sickle cell anemia had a different type of hemoglobin than normal people. And let's have a look and see how this works out. The parents of the people with the sickle cell anemia had a small proportion of cells that sickled, but they had a mixture of normal and the abnormal sickle cell hemoglobin. So two parents, each of which had a mixture of the normal and the abnormal. Now, there's a gene for the normal and a gene for the abnormal hemoglobin. So each parent contributes one of its genes to the offspring. If both of those genes are the abnormal genes, then you have an individual who makes only the abnormal, the sickle hemoglobin. If both of those are normal genes, then you have an individual who makes only normal hemoglobin. But if one of them is a normal gene and the other is abnormal, then you have a carrier just like the parents. So you can get three combinations from the carriers: the abnormal, the mixture, and the fully normal. And the rules of inheritance, the fact that there's a chance of about half of passing on one of these genes and a half of passing on the other genes, those rules tell us that we should expect about a quarter of the diseased, a half of the carriers, and a quarter of the normal in such a mating. That's one of the most fundamental of the Mendelian ratios. Now, we've got a little experiment here to show you how the statistics of those ratios work out. Let's have a couple of volunteers. There we are, a couple from, yes, let's have you, and let's have you. Yes, the girl in the red sweater. What's your name?
Emma.
Emma. And yours?
John.
John. So Emma and John are going to act as if they're a mother and a father. Now, here we've got the abnormal hemoglobin. Those are the abnormal genes. Now, why don't you put those into there, Emma? Shall we put them into that? Just put them into there, right? Now, you take hold of the others. Put those into there, right? So now we've got a mixture. Did you see equal numbers of the two sorts of genes? Let's put your hand in there. Let's get them all, shake them all up. Shake them all up so they're nice and jam. Huh? Now I want you each to pick out one ball. Pick out one ball. Don't look. Pick out one ball. Now let's see. You've got a blue and a red. I put the blue and the red in the middle. Okay? Now keep doing that. Pick out a ball each, and if it's a blue and a red, put it in the middle. In the middle. If it's two blues, put it in this end, and if it's two reds, put it in the other end. Come on, another one. One each. Okay, that goes in that end. Okay. Again, let's keep it going. Let's keep it until we've got a reasonable idea. Okay, in the middle, a blue and a red in the middle, and another one, another pair each. Okay. There we are, in the middle again. And another pair. Let's put them in. Blue and a red. The blues and the reds are winning, aren't they? Have another go. Come on, another go. Oh, finally, a blue and a blue. Another go. Keep going. Okay, a blue and a red in the middle. Blue and a red together in the middle. Keep going. There we are, red and a red in the end. Let's have another go. Until we fill up the middle one. Blue and a blue in this end. Okay, and another, just one last one. Okay. Red and a red in the other end. You see how we've got it? About 1 to 2 to 1. Just to show you how the Mendelian ratios work. Thank you very much, both of you. So that's the fundamental pattern of inheritance. But what actually has gone wrong with that hemoglobin molecule? What's gone wrong with it is that in one position, one of those amino acids has changed. Now we've got here a computer graphic of a hemoglobin molecule. You see it coming up here, and you see the chain, that's the basic chain of amino acids that gives the hemoglobin its structure. And you'll see here we're moving in to one position, the sixth position, and that's a glutamic acid. Glutamic acid there in the normal hemoglobin. See it switch? That's a valine. It's that one little difference between the normal hemoglobin and the abnormal hemoglobin that gives all the problems. So let's think of that in terms of our DNA code. Glutamic acid. The code for glutamic acid, the triplet code for glutamic acid, is GAG. So that's the situation in the normal hemoglobin. What happens in the sickle cell hemoglobin? There's been a switch from A to T, from glutamic acid to valine. It's that one little change from glutamic acid, A to T to valine, that causes all the problems. That's what causes the disease. Now let's think of that for a moment. How much DNA is there that goes up to make our genetic information? How many of those letters are there in the total DNA content in the cell? Well, there are an awful lot of them. 3,000 million letters. Just one of them has gone wrong. Just one little misprint. Now, let's think of that. That number of letters is just about the number of letters that you'd find in 100 Encyclopedia Britannicas. And we've got them all here for you. 4 sets of Encyclopedia Britannica. And somewhere in one of them, we've got a misprint. Now, just have a look very carefully. Have a look. Can you see the misprint there? Anybody see the misprint? I've got it here in the book here, in the first volume. Anybody seen it? Yes. What is it? Tragedy. Yes, it's wrong, isn't it? It's a misprint in the Encyclopedia Britannica. See, there you see it, written in large. Just one letter gone wrong in all those 3,000 million. That's what causes the disease. Now, next time, I'm going to tell you how the genetic engineers can find out where that letter is and how it's gone wrong. Thank you.